This is Earth, a place that you and I call home, a place you and I were born, and a place we will spend all of our lives. This would also be true for our next generation and the generation after that. Maybe. In the previous century, we began taking steps towards sustaining life outside of Earth, specifically outside of its atmosphere and gravity. In 1961, Yuri Gagarin spent 108 minutes in space while orbiting the Earth. In 1969, Neil Armstrong, along with the Apollo 11 crew, spent 8 days in space on their journey to the moon and back. Finally, Valery Polyakov in 1994 spent 437 days in space on board the Mir space station. In each instance, we pushed the limit of our stay in space. But why? Because the ultimate goal of human space exploration is to travel to distant worlds. And this is going to require us to be outside of Earth's atmosphere and gravity for a very, very long time. Much, much longer than cosmonaut Polyakov. How this will affect our body is the main reason Polyakov stayed in space so long. But that time period was not enough to understand all of the effects this has on the human body. So scientists do what they do best when a problem cannot be studied directly. They create or find the best available model of the problem. Experiment on that model to find possible solutions, then apply those solutions to the problem. Creating or finding these models is paramount if we ever plan to travel to distant worlds in the celestial sphere. Life, since its inception on Earth about 4 billion years ago, has been under the influence of Earth's gravitational force, a force that accelerates everything that's free to move towards the ground at 9.8 meters per second squared. As life evolved and diversified, the gravitational force that affected these developments remained unchanged. Even as millions of species changed the very environment that they were in, including the entire atmosphere itself, gravity remained the same. Put it simply, life on Earth is optimized for functioning with a gravitational acceleration of 9.8 meters per second squared. So what happens when we move a human body to an environment where the gravity is less? One of the most notable effects is bone loss. This happens because the lower gravity will put less physical stress on the body, and this in turn will prevent it from building stronger bones to counteract this lesser stress, thus making them weaker. Similarly, muscles will become weaker, especially the leg muscles. Body fluids are redistributed more evenly throughout the body because they're no longer pulled down towards the legs. The heart doesn't work as hard because it's no longer working against gravity to pump fluids around, so it too becomes weaker. Now, on the other side of the spectrum, very little research has been done on the long-term effect of stronger gravity on the human body. In gravity stronger than 1G, stronger bones would be built, but this might cause other issues with the body. The only way we can simulate stronger gravity is by putting the subject in a giant spinning centrifuge. This is a complex task for long-term studies because an entire livable room has to be created with everything that a person would normally need from day to day, including resupply while the room is constantly rotating. Even if that could be done, they would still be directly affected by Earth's 1G gravity. For this reason, most of our non-1G human research is done in a microgravity environment because we already have 2 space stations orbiting the Earth. But even with our 2 space stations, the long-term effect, 10 plus years, on the human body that low gravity and increased radiation has, is difficult to study. The key to partially overcoming this difficulty is to create or find a physical model that closely responds to changes in gravity and increased radiation similarly to the parts of the human body that we're interested in studying. We can create a software biological model that simulates certain aspects of the human body, like genes and proteins. We can also create biomimetics devices. These devices physically emulate certain aspects of the human body. Aspects such as blood flow through the heart can be modeled with these kinds of devices using actual fluids. But sometimes not enough prior data is available to create a reliable model in the first place, or we are concerned with how an environment affects an entire organism. In cases like these, using another living organism as a model may be the only solution for acquiring a useful model. For modeling aspects of the human body, many times other living mammals are used. While in many cases these mammals represent the most accurate models, depending on how they are used can raise issues about ethics. Ethics becomes less of an issue as the organisms that's used as a model becomes physically smaller and smaller, and especially when they're considered to be pests. Of course, the smaller an organism is relative to humans, the lower the chances that its internal organs will be similar to humans. But still, there are some small organisms that are remarkably similar to humans when comparing the digestive system, heart, other organs, and DNA. The fruit fly is one of those organisms. The fruit fly, scientific name is Drosophila melanogaster, is of the phylum Arthropoda and class Insecta. Fruit flies most likely originated from sub-Saharan Africa about 10 to 15 million years ago. About 12,000 years ago, the fly migrated out of Africa along with migrating humans to other continents. Today, fruit flies are found on every continent except Antarctica. What makes fruit flies so attractive as a biological model for humans are the following. Fruit flies have complex organs and systems like the brain, heart, kidney, and an immune system. They use highly similar genes and molecular pathways for fundamental processes like cell division and cell signaling. About 75% of human disease-causing genes have a functional equivalent in the fruit fly genome. While these facts about the fruit flies makes them a good biological model for humans, that's only part of the requirement of a good model. For a model to be good, it also needs to be practical. That is, it needs to be easy to use when setting up and conducting experiments. To that end, fruit flies are very practical. Its care and culture requires little equipment, space, and expense, even when using large cultures. It can be safely and readily anesthetized, usually with ether, carbon dioxide gas, by cooling. Without anesthetizing, it is very difficult to work with flies. There's a commercial product called FlyNap that immobilizes these flies for up to several hours. FlyNap. FlyNap. That's an interesting name. In any case, its morphology is easy to identify once anesthetized. It has a short generational time, about 10 days at room temperature, so several generations can be studied within a few weeks. It has a high rate of egg production. Females lay up to 100 eggs per day and perhaps 2,000 in a lifetime. The development of this organism from fertilized egg to mature adult is well understood. All of this makes the fruit fly a very good biological model for studying humans, especially in an environment that's not readily available, something like a less than 1G environment. The only way we can create such an environment that's stable over a long period is an orbiting space station. Both the International Space Station and the Tiangong Space Station have special facilities to accommodate fruit fly experiments. Even though these experiments might be simple to conduct on Earth, in space, it is much more difficult because of the logistics of getting them there, conducting the experiments while there, and finally return the samples back to Earth. There are many aspects about the life cycle of fruit flies in the space environment that interest scientists. Some of them are, how does the immune system and pathogen interactions function in microgravity? How does the cardiovascular system function in microgravity? What are the effects of long-term spaceflight on development, aging, and behavior? What are the effects of long-term spaceflight on gene and protein expression? To try to answer these important questions, a principal investigator or lead scientist has to first choose one of the two space stations, Tiangong Space Station, TSS, or the International Space Station, ISS. These are very expensive and advanced pieces of hardware that so far only governments are building. And because of this, a principal investigator may have only one space option available. In this video, we look at the experimental facilities that NASA has used in the past and present on the ISS. More information about the fruit fly experiments on board the ISS is available online than the fruit fly experiments on board the TSS online in English. NASA uses the fruit fly laboratories on board the ISS to conduct experiments on fruit flies. The laboratory is based on three main elements: assembly of cassettes, food changeout platforms, centrifuge, and microgravity container. The cassette is a rectangular box that has a section that allows a tray to be inserted in it. The tray contains the food for the flies. The food is specifically made into a gel to prevent it from floating around in the micro-G environment of the ISS. The food tray section effectively divides the cassette into two sections. The empty section without the tray is where the flies are initially housed. The flies are not adults yet. Instead, they're pupae, the final development stage of a fly before they become adult flies, the ones we are used to seeing. This is done for two main reasons. The first reason is that the pupae have a hard shell. The adult fly doesn't. This helps the flies to withstand the G-forces associated with launching a rocket. This is typically 3 Gs. The second reason that the flies are launched in the pupae stage is to make sure that they are not adults yet until they're on board the ISS and the station's crew is ready to initiate the experiment. This ensures that the adult flies will never experience normal gravity. Each cassette can hold flies for independent experiment. For example, one cassette can hold normal fly food to study generational changes in the fly, while another cassette can hold fly food infected with pathogens to see how the immune system of the fly fights the infection in micro-G. Each cassette is loaded into a small case called observation unit. It contains a digital camera and an LED light. The camera allows the researchers on Earth to monitor the progress of the fly in the cassette. The LED light is used to simulate day-night cycles. Fruit flies have a circadian rhythm. Circadian rhythm is roughly a 24-hour cycle that influences certain aspects like sleep, testosterone secretion, blood pressure, and more of an organism. Within this cycle, certain aspects of the body are influenced at a specific time offset. Although the timing is controlled by an internal biological clock-like mechanism, it can be affected by external input like light from natural or artificial sources. Fruit flies and humans have circadian rhythms, so one type of fruit fly experiment conducted is to change the day-night cycle to something other than 24 hours and then note the effect it has on the circadian rhythm of the fly in a micro-G environment compared with a control group of flies on Earth. Real-time data about the environment in the cassette, like light intensity, temperature, and humidity, are transmitted to Earth. This data can then be used to control the environment of the flies that are part of the control group. The container that houses the cassette, LED light, and camera is normally in a micro-G environment like the rest of the ISS. But like what was stated at the beginning, one of the main questions that scientists are interested in is how gravity at different strengths affect the human body in the long term. To begin answering these questions, a special compact centrifuge is placed next to the 18 receptacles that accepts an observation unit. The centrifuge also accepts 4 observation units. As the centrifuge spins, the centrifugal force will create simulated gravitational force. The strength of this gravitational force is determined by how fast the centrifuge spins. Scientists control the spin rate to change the simulated gravity. While this centrifuge is used to simulate gravity on the Moon or Mars, it is mostly used to simulate the gravity of Earth. Why is that? Well, when scientists run fruit fly experiments on the ISS, the flies are not only exposed to the microgene environment, but they're also exposed to external radiation like galactic cosmic rays and solar energetic particles. These are high-energy particles originating from the sun. These radiation will also have an effect on the flies, along with the effect of being in the microgene environment. So to distinguish between the effects caused by radiation from the effects caused by the microgene environment, observation units are placed in a centrifuge and spun to create normal Earth gravity. Effects of the radiation will affect flies in the observation unit, in the receptacles, and the observation units in the centrifuge in an identical way. However, the effect due to microgravity will only affect the flies in the observation units in the receptacle, and not the ones in the centrifuge. Now, revisiting the experiments with the fruit flies that are infected with pathogens, after a certain amount of time in the microgene environment, they are now ready to be studied and analyzed. However, the ISS is not an analytical lab. Everything in the space station is limited, including electrical power and, ironically, space itself. For this reason, many of the scientific devices are limited to only exposing and maintaining specimens in a micro-G and elevated radiation environment. Comprehensive analysis is done back on Earth when the specimens are returned. This is one reason why a sample return mission to Mars is an important mission, because it will extend our knowledge beyond the Mars rocks and soil that will be returned. We will be able to analyze the containers that the samples were in and see how they were affected by being on the surface of Mars for many years using the best equipment available on Earth. But back to our infected fruit flies. When the experiment is over, the most important immediate step afterwards is to fix the flies. That is, whatever state they're in needs to be preserved. If this is not done before the flies are returned to Earth, there's a very good chance that reintroduction to the Earth's surface environment and gravity will alter the state of the flies and possibly ruin the experiment. There are two ways to fix the flies. One is through the use of special chemicals that first kills the flies and then protects certain aspects of its cells from degrading. The other way of fixing the flies is through the more familiar method of freezing. The ISS has multiple freezers on board capable of freezing the fruit fly cassette within a minute. The cassette will remain in the freezer until it's time for the return trip to Earth. It's then transferred to a cold storage on board the return capsule. This is a portable freezer. After touchdown, it's yet again transferred to another portable freezer or dry ice box on board a helicopter that will then fly it to a designated nearby lab for the proper analysis to be conducted. The entire process needs to be planned carefully, and while on board the space station, it should require the least amount of time from the astronauts. Keep in mind, they have many other jobs to do on the station, so therefore each experiment that they set up, manage, and break down needs to require the least amount of their time as possible. So in summary, here are some of the effects caused by microgravity on fruit flies. 1. Compromised immune systems. Flies were less effective at fighting diseases. 2. High molecular damage to the cell. This contributes to accelerated aging. And 3. High rate of anomalies for flies that went through the entire development stages in micro-G, that is, from embryo to adult flies. All of these effects, if applicable to humans, can severely affect the ability for humans to stay in space for a very long time. Long time in terms of multiple years. Having a research station on Mars would require humans to stay in space for at least 3 years. Going further still, to the moons of Jupiter and Saturn would take decades. The further we move away from Earth, the more self-sufficient we'll have to become. The more independent of Earth we'll have to become. Then, at some point, Our biggest challenge may not be the logistics of creating the materials we need to survive. It will be the challenge of a human embryo developing into a normal human. I'm Dex DFX for the Celestial Sphere.